Hello students, in our previous sessions, we have learned types of reproduction, structure of flower, development of male and female gametophyte, types of pollination and pollinating agents, contrivances for cross-pollination and outbreeding devices from chapter number 1, reproduction in lower and higher plants. In this session, we are going to learn three topics. The first one is double fertilization. The second one is development of endosperm. And the third one is development of embryo. Double fertilization is the important step in the process of sexual reproduction in angiosperms. The process was first discovered by a scientist called Nauschiel in lilium plants. Double fertilization is a characteristic feature found in angiosperms in which both the male gametes are used. The pollen grains, after pollination, are deposited on the surface of the stigma. The pollen grains absorb sugary substance for their germination secreted by the surface of the stigma. The pollen grains on germination form a pollen tube on the stigma which pass through the style and enter the ovule mostly through the micropyle. In the first diagram, we see that the pollen tube enters through the micropyle. In the diagram A, we can clearly see that the pollen tube enters through the micropyle. Hence, the phenomenon is called as porogamy. In the second diagram, the pollen tube enters through the chalaza and it is called as chalazogamy. In the third diagram, we see that the pollen tube enters through the ovule by piercing the integuments. This phenomenon is called as mesogamy. The pollen tube is hollow and carries two non-motile male gametes. Hence, the phenomenon is called as siphonogamy. The process of fertilization occurs inside the embryo sac, or it is also called as female gametophyte of angiosperm. The pollen tube carrying male gametes enter one of the synergid in the embryo sac. The pollen tube absorbs watery content from the synergid and ruptures or breaks, and it releases male gametes inside the embryo sac. In angiosperms, as there are two male gametes for the process of fertilization, fertilization occurs twice at two different places. Hence, it is called as double fertilization. The diagram shows the process of double fertilization. You know that after pollination, the pollen grains are deposited on the surface of stigma. The pollen grains absorb sugary substance from the surface of the stigma and pollen grains germinate, producing pollen tube. The pollen tube passes through the style, then reaches to the ovule and enters inside the ovule. The pollen tube enters by different methods. We have seen that there are three different methods depending upon the entry of pollen tube. The entry of pollen tube through the micropyle is called as porogamy. The entry of pollen tube through the chalaza is called as chalazogamy. and the entry of the pollen tube by piercing the integument is called as mesogamy. So, in angiosperm, the male gametes are released inside the embryo sac. The first haploid male gamete fuses with haploid egg cell or female gamete, and it will produce a cell called as zygote. As two haploid gametes unite, that is haploid male gamete and haploid female gamete, the cell produced, that is zygote, is diploid in condition. This phenomenon or process is called as syngamy or first fertilization. The zygote later on develops into an embryo inside the ovule. The second male gamete, which is haploid in condition, comes at the center of the embryo sac and fuses with diploid secondary nucleus. This process is called as second fertilization or triple fusion, and it produces a triploid cell called as primary endosperm nucleus. Triple fusion means that three haploid nuclei, two from secondary nucleus and one from male gamete, fuse with each other. The triploid primary endosperm nucleus later on develops into an endosperm, a nutritive tissue which provides nutrients to the developing embryo. In this way, the process of double fertilization occurs inside the embryo sac in angiosperms. Now we will study the significance of double fertilization in angiosperms. Double fertilization is the characteristic or unique feature of angiosperm. It means it is only found in angiospermic plants. The diploid zygote after cell division, that is mitosis, develops into an embryo of the seed. On seed germination, the embryo develops into a new plant. The triploid primary endosperm nucleus undergoes mitosis to form an endosperm. Endosperm is a nutritive tissue which provides nutrients to the developing embryo. The diploid condition in angiosperms is restored by the fusion of haploid male gamete and haploid female gamete. Double fertilization helps to avoid polyembryony, that is, formation of more than one embryo. Now we will study post-fertilization changes. Post-fertilization changes are the changes which occur in the ovary and the ovule after fertilization. Hence, they are called as post-fertilization changes. First, we will study the development of an endosperm. It is one of the post-fertilization event. The triploid primary endosperm nucleus undergoes repeated mitotic divisions to produce an endosperm inside the embryo sac. Endosperm is a nutritive tissue which provides nutrients for the developing embryo. The other cells of the embryo sac get disorganized or degenerated. Depending on the mode of development, there are three types of endosperm. The first one is nuclear endosperm. In the diagram, we see the zygote develops a wall around it and is called as oospore, and simultaneously primary endosperm nucleus develops into an endosperm The primary endosperm nucleus undergoes free nuclear divisions which occur in the nucleus of triploid primary endosperm. Initially, only nuclear divisions occur, and later on there is formation of cell wall. At the center, we see presence of a large vacuole surrounded by number of nuclei. In the last diagram, we see that after the formation of nuclei, around each nucleus, a cell wall is produced. So, in this way, initially, in nuclear type of endosperm, only nuclei are formed, and after the formation of nuclei, cell wall is developed around each nucleus. So this type of development of endosperm is called as nuclear endosperm. The common examples of nuclear endosperm are coconut, wheat and sunflower. The second type of endosperm is cellular endosperm. In the diagram, we see that there is division of primary endosperm nucleus. In the first diagram, we see that there is division of primary endosperm nucleus into two cells. Then in these cells, the nucleus also undergoes division. Similarly, number of free nuclear division occurs inside the primary endosperm nucleus. Similarly, there is formation of cell wall around the nucleus. On the upper side, we see the development of oospore into an embryo. And in the last diagram, we see that number of cells are developed inside the embryo sac. During the cell division, the nucleus as well as cell wall is developed simultaneously. Hence, it is called as cellular endosperm. The next type of endosperm is called as halobial endosperm. The diagram shows that the first nuclear division of primary endosperm nucleus occurs by the formation of two unequal cells. The upper cell is large and it is present towards the micropylar end, while the lower cell is small and it is present towards the chalazal end. So initially, primary endosperm nucleus undergoes division and produces two unequal cells, larger cell and smaller cell. Now in each cell, the nuclei undergo divisions and later on cell wall is developed around each nucleus. The example is asphodylus, and the examples of cellular endosperm are petunia and adoxa. So we have studied the development of endosperm. In this topic, we are going to study development of an embryo. Embryogenesis is the process of development of zygote into an embryo. The process of development of embryo in dicot and monocot is same. Only the difference is that dicot embryo shows development of two cotyledons, while monocot embryo shows development of only one cotyledon. In the diagram, we see development of monocot embryo. In the A diagram, we see oospore. Oospore means zygote develop a wall around itself. Hence, it is called as oospore. The oospore divides into two-celled structure called as proembryo. We can see it in the diagram B. It shows upper large cell called basal or suspensor cell at the micropylar end and smaller cell called as embryonal initial towards the chalazal end. Further, mitosis occurs in suspensor initial as well as embryonal initial. In the C and D diagram, we see that suspensor initial and embryonal initial cells undergo mitosis. In the E diagram, we see the suspensor cell has divided and it produces suspensor. The first cell of the suspensor towards the micropylar end becomes swollen and enlarged in size and functions as haustorium. Haustorium helps to absorb nutrients from the developing endosperm for the development of embryo. The lowermost cell of suspensor is called as hypophysis. The suspensor helps in pushing the embryo into the endosperm. The embryonal initial undergoes three successive divisions to form octant stage. The planes of the cell division are at right angles to each other. In the next diagram, that is F diagram, we see that the lower tier of four cells of octant give rise to hypocotyl, whereas radical is also formed. Then four cells of the upper tier form plumule and one cotyledon in monocot and two cotyledon in dicotyledonous plant. In the G diagram, we see further development of embryo. Later on, the embryo becomes curved and appears as horseshoe shaped. In the last H diagram, we see seed of monocot. The seed of monocot at the top shows presence of single cotyledon. You know that monocot show presence of only one cotyledon. This cotyledon is called as scutellum. Then at the lower side, coleptile is present. Below the scutellum, coleptile is present, and inside the coleptile, plumule is present. So the function of coleptile is to protect the plumule. Later on, plumule develops into shoot apex. Then epiblast is present at the center. The radical is at the lower side and the radical is protected by a tissue or a covering called as coleryza. Then this radical later on develops a structure called as a root cap. So in this way, the development of embryo occurs after fertilization. And finally, ovule is transformed into the seed and ovary develops or it is transformed into the fruit. So in this session, we have learned double fertilization, development of an endosperm and embryo. Remaining part we will see in our next session.